Now we're going to discuss phenotype prediction BUGBASE, and which is used to predict bacterial phenotype database based on I16S OTU table, and the official website is given here. The article entitled with BUGBASE predicted organism level microbiome phenotypes, published on the preprint magazine in 2017, and has been cited so far more than 371 times in research articles. BUGBASE is a tool for analyzing the phenotype of microbiome sample, and this website can predict and compare a large amount of information based on OTU table and mapping file, including gram-positive, gram-negative, biofilm forming, pathogenic potential, mobile element containing, oxygen utilizing, oxidative stress tolerant. And the BUGBASE workflow diagram is just like this, that pre-existed databases they use, such as usearch, BUGBASE, Pictrust, and predicted trace per OTU, and the input file required OTU table and mapping file. And when we run BUGBASE, ultimately we generate 16S normalized OTU table, threshold, OTU contribution, predicted phenotype, and we get such kind of plots. The requirement for this, we need OTU table and mapping file, and the standard format of OTU table biome format. For 16S use green genes, and for metagenome we need IMG as a reference. And mapping file should be tab separated. The first line must start with sample ID, and first line must be column header. The first column must be sample ID, and only letters, number, underscore, and hyphen are allowed. You can see here. Again, there are several ways to perform this analysis. We can perform under run locally on window by executing this code with specify the first software directory as the BUGBASE variable in pipeline. Enter the GGOTU table, metadata, specify the group column name, and output directory. You can see here. And the second method is we can use ImageJP online server. We can go to the BUGBASE section and we copy and paste our data, and we can execute the plot button, and then it generate our result files. And the third is installation and use in Linux system, which is totally optional. So in order to run Linux server, we have to switch our terminal from Git bash to Windows subsystem for Linux for Windows user. And then we have to install some software, for example, git clone. And then we have to install dependencies package BUGBASE, and then we have to test data. And then we will continue by running the code step by step. We can set parameter and run. You can see here, export BUGBASE path, export this, run BUGBASE, GGOTU tab text, mapping file. And the fourth method is also available by visiting the website, the official website of BUGBASE, and then we can use this by clicking on the parse data. We can upload our OTU tab file, and we can upload mapping file there, and then we can choose the column header fill in group, and then we will just wait to generate our result. And normally the result file include normalized OTUS, which contains 16S normalized OTUS text, OTU contribution, which have aerobic, anaerobic, stress tolerant PDF. Each group of functional gate is horizontally composed of state diagram and legends contributing OTU's text, each OTU corresponding to the presence or absence metric of each type of function. Predict phenotypes, which is relative abundance of phenotype. We get here aerobic, anaerobic, stress tolerant PDF, and box plot for each site of feature, and threshold, which is the annotated abundance at different threshold, contain anaerobic, aerobic, stress tolerant PDF with line chart of classification, proportion, and relative abundance in different method. Now let's have a look and try the window run, and we will use RStudio. We go back and we will execute the code. Now we are in the section of BUGBASE bacterial phenotype prediction. We're gonna run these commands one by one. Now see BUGBASE command line analysis. We will run this, and the script has been optimized for R4 version, and the biome package has been updated to biome format. And then we will execute this code. Now see, it's processing here. It takes around just few moments to finish. Then we will see here the files generated in the folder of BUGBASE. Now see, we have here normalized OTUS, we have OTU contribution, we have predicted phenotype, and we have threshold. Let's have a look. We come here and we just see BUGBASE and we will see to each. Now see, it contains 16s normalized OTUS text. It contains OTU contribution. We can see here, this is anaerobic PDF. This is anaerobic. This is for contain mobile element. And this is stress tolerant, for example. And this is text legend PDF. You can see here. And in the next folder, we have predicted phenotype, aerobic. We see here, we have anaerobic PDF. And it also contains some stats text file as well. We can see here facultatively anaerobic PDF. And we have potentially pathogenic PDF. You can see here. And under threshold folder, we have aerobic, anaerobic, facultative. You can see. So all these files we generate by using the command line analysis in RStudio. Now we can see that how we can use ImageJP to run BUGBASE analysis. This is our ImageJP. We just go to the BUGBASE section. Now we have to paste over OTU.txt file here, which is already saved here by default. And there is sample group file with first column as a sample name and other column as a sample attributes. We have here, you can see our KO, OE and WT. It is adjusted by default here. So we just choose here group and then we click plot, and then it takes around a few moments to finish. Now we get here download result file, click over here. We have this result BUGBASE zip. We can just copy this and paste it in the same folder so we can see what's the difference. So we will extract it first. Now again we receive the same 4 files, the folder normalized OTUS, OTU contribution, predicted phenotype and threshold. So whatever method we use, we will get this kind of result. And so we prefer to use ImageJP, and we can run analysis under local command line mode instead of using Linux system. And if you want to use, you can use it. You can go to the pipeline and you can just go to the section appendix. Now see appendix analysis under Linux server, and you can choose here BUGBASE, and then you have to install software, you have to install dependencies, and then you can execute the code one by one, and then you will generate eventually all these folder which we discussed earlier.